University of South Carolina, an MS in biology from Virginia Commonwealth University, and a PhD in immunology from the University of Kentucky. After postdoctoral fellowships at Johns Hopkins and NIH, he became a principal investigator at NIH in immunology. He later joined the ECU School of Medicine to pursue both research and teaching. In 2000, he transitioned to high school science education at Knoxville Catholic High School. Dr. Kearse studies the Shroud of Turin, focusing on the properties of its bloodstains as a subject bridging faith and science. Let's bring up Dr. Kearse.
Okay, go. Okay. Thank you. About 13 years ago, I wrote an article for Barry's website. It was called The Blood on the Shroud of Turin, an immunological review. So in this first part, what I'd like to do is just kind of bring that up to date, where I think things sit in 2025. In the second part, I'm going to talk about blood transfer. So here are kind of the three main characteristics that you usually hear about blood on the shroud, that scientists have studied it and they've determined it to be human, to be blood from a male, and to be type AB. So we're going to go through each of these individually. For the human designation, you have two options there. You can go a DNA route, or you could go an immunological route using antibodies. For sex determination, that is, you use DNA methods for that. I'm actually going to start with that one. I'm going to begin by talking real briefly about the technique that's used for this, because I think that's important to help understand the results. The technique is called polymerase chain reaction, or PCR. So here we have all of the genes within a nucleus that are necessary to encode a human being. So the human genome consists of about 20,000 to 30,000 total genes. What PCR does, you make a probe to one of those specific genes that you're interested in. It will go in among those 20,000 to 30,000. It will locate it. It will not only locate it, it will copy or amplify it in a chain reaction fashion, which is why this is called polymerase chain reaction. It's so efficient at copying that you can get about 1 billion copies in just 3 hours' time. So if you have a situation where you don't have much DNA, through PCR you can amplify that up to get enough useful material to look at. So here's a real-life example of this. So this is a study we did early last year related to Eucharistic miracles. So these are wheat wafers that have been baked at a very high temperature, which is not particularly DNA friendly. So you expect the DNA to be very degraded, very fragmented, and you can see, in fact, when you run it out on a gel, you really don't see anything at all. But if you take the same material, we went in for a probe with one particular gene we were interested in and did PCR, and we get a useful signal. So we expected the gene to come out where the arrow is, that's where it is. And then what we do is take that material and sequence it. That verifies that it's the particular gene that we're interested in. So even in situations where the DNA is in really bad shape, and it's extremely degraded, extremely fragmented, through PCR you can often get enough useful material to look at. And a lot of times you don't really even need the full-length gene to do this. A lot of times you can get even just a small piece of it, and that's sufficient. Now, as far as the shroud goes, what's been done on the bloodstained fibers for sex determination, there were two studies that came out, one in the mid-90s and one in the late 1990s. Probably the most famous is from the book, The DNA of God by Dr. Garza Valdez. What he did, he used PCR to look at 3 specific genes. So he looked at the X and Y for sex determination. He also looked at a portion of the hemoglobin gene, the beta globin gene. He got a positive signal for all 3. So he concluded from this that this shows that the blood on the shroud is from a human male. And he said that he knew that the DNA signal was coming from the blood cells because he got a positive result for the beta globin gene. Unfortunately, what he overlooked is that the beta globin gene is also present in skin cells. So there's no evidence in this that the DNA signal is truly coming from blood cells. There was another report that came out in a Shroud magazine about mid-1995, mid-90s. There, when they looked at it, they found both male and female DNA present. Also, when they looked for specific markers unique to individuals, they could account for the profiles of at least 6 different persons. So clearly there was a contamination here. The results were considered scientifically null and void. Now, I've written about this. Here's the reference for it. If you're interested in that, you can go back to this slide and just look it up. Or you could just type my last name into a search engine and it should pop up. As far as human species determination, no one has really done the DNA route for the shroud. One of the reasons is it always gets tough. Well, do you know if the DNA signal is really coming from the blood cells or could it be coming from the skin cells? One way you could get around this is to look at immune receptor genes. So immune receptors are expressed only on blood cells, one particular type called lymphocytes. Both blood cells and skin cells have immune receptor genes. But what's unique about this, in the blood cells, the DNA is literally cut, spliced, and pasted together. The DNA undergoes rearrangement in blood cells. They're the only cell types in the body that do this. So you can make a probe to the rearranged form. It will find that form and ignore everything else. So in this case, it would not matter how many hundreds of people, how many thousands of people, how many hundreds of thousands of people might have touched the shroud throughout its history. The probe would completely ignore it. So if you got a signal from that, you could then take that, sequence it, you could get some potentially useful species determination. Now this has not been done related to the shroud. It's mentioned in the same article that I talked about just a minute ago, if you're interested in that. What has been done as far as species determination in the shroud, there have been studies using serological methods, using antibodies. These were done in the early 80s by 2 groups, an Italian scientist, Bima Bologna, and then Heller and Adler, who studied samples collected by the original STIRP team. They looked at 2 major blood proteins, immunoglobulin and albumin. The results were essentially the same, so I'm just going to talk about these collectively. So they took anti-human antibodies to human blood products. And antibodies themselves are very, very small. You really can't see them with the naked eye. So typically what's done is you attach some kind of fluorescent tag or chemical tag to them. And then you can look in the microscope, and wherever you see this light up, you know that the antibody has found its particular target. So they took unstained fibers from the shroud without blood, blood-stained fibers from the shroud. They reacted them with these anti-human antibodies labeled with a fluorescent tag. And here's what they found. The control fibers were negative. The blood-stained fibers were positive. So they concluded from this, human blood components are present in the blood-stained fibers of the shroud. Now, an immunologist is going to look at this and they're going to say, well, I think you've shown that blood components are present in these fibers, but I'm not really convinced that you've demonstrated that they're human blood components. And this is why they're going to say that. When you have these anti-human antibodies that you're using, something that's often not even appreciated by those who use these, what this anti-human designation means, that refers to how the antibody was made, but not necessarily its reactivity. What all this anti-human really means, someone took a human protein, they put it into another species, and that species made antibodies against it, but it does not necessarily comment on its reactivity. In immunology, there's something called cross-reactivity. You can make an antibody against a protein in one species, but it may in fact react with the same or similar protein in other species, and this is in fact the case. Now, Bima Bologna, in his studies, he never really addressed cross-reactivity, which was a pretty major oversight. Heller and Adler were very well aware of cross-reactivity, so they did some experiments to look at this. So Heller and Adler took their anti-human antibody. They reacted it with blood products from humans. It was positive. They also found that it reacted with blood of primates. So their original conclusion by Heller and Adler was really, this data shows that the blood is at least of primate origin. They never really said human blood. It just kind of, through discussion of others through the years, it just kind of morphed into that. They also showed when they reacted with blood from cow and from horse, it was negative. What we now know, though, some 40 years later in hindsight, the cross-reactivity is in fact much greater than was originally considered. We now know that these anti-human antibodies would also react with blood from rodents, from cats, from dogs, and various other species. So the conclusion from this is a reference for it if you're interested in looking that up. I'll just read the highlighted parts. As discussed, cross-reactivity precludes a definitive assignment of human or even primate blood being present on the shroud. The blood is most correctly classified as species unknown. Now that's going to bring us to MNS. So these are three separate molecules expressed on red blood cells, M, N, and S. The S is the most important one. Around the mid-'80s, Bima Bologna reported in the Shroud magazine that Shroud bloodstained fibers were positive for M, N, and S. Now, it was a short article. In fact, he never really even showed in it any results for anti-S reactivity. All he said was it was just a written description of fairly good binding. Exactly what that means, who knows, but it's potentially interesting because Kearse, pointed out about in 2012 that S antigens are specific for humans. So this certainly has some potential interest. However, there's more recent data that shows that anti-S antibody preparations that would have been used at this time, they're really not suitable for species determination. And here's how that was revealed. In certain xenotransplantation studies, what that means, you take an organ from one species and you transplant it into another. So as a way to kind of deal with the organ shortage, they tried to take organs from pigs and to put those into humans. They were rejected quite quickly. So as that was worked out, what it was elucidated is that human anisura just naturally contain antibodies that will react with white blood cells from many non-human species, not just pigs, but pretty much everything across the board. So these type of anti-S preparations, they're not useful for species determination. I'm kind of looking back on it, it's probably the worst thing that you could do for that. So here's the reference for that. I'll just hit the highlight parts. Such anisura were particularly prone to cross-reactivity with blood from a bountiful number of other species. These findings underscore the necessity of using more current serological tools, that just means antibodies, in any future investigation of bloodstained artifacts such as the Shroud, particularly in relation to species determination. That's going to bring us to type AB. So I'm not going to talk a lot about this because I've talked about it quite a bit before. But in the early 80s, Bima Bologna reported in Shroud magazine that the Shroud bloodstained fibers were type AB. So here's a geographical or a population distribution of blood type. There's going to be some variation depending on geographical location, but pretty much this is how it rolls. Most people are type O. A close second place is type A. A pretty distant third is type B. Rarest of all is AB. It's usually around 3 to 5% in the population. Some variation depending on geographical region. Sometimes you'll hear, well, in the Middle Eastern population or Jewish populations it's 20% or so higher, not based on any solid scientific data. So we have this rare blood type AB. It's been described for the shroud. It's been described for the siderium. It's been described for the tunic of Argentul as a proposed robe that Christ wore on the way to Calvary. Also, various Eucharistic miracles that have been typed to date. All of these show up as AB. So it's been said, well, this is a statistical impossibility. This could happen by chance. Odds greater than 3.2 million to 1. Some even put this in the billion to 1 range. A recent article in Catholic Answers magazine said this, well, this is mathematical proof of the real presence, a proof that God is real, proof that our Lord has AB blood. If you look at what the structures of the A and B molecules are, they are made of carbohydrates, or in other words, sugars. And the main part of the chain is exactly the same in A and B. What makes it different is there's a different sugar that's added to the side of A than there is B. So that's really the only difference between A and B, is which particular sugar side chain that there is. Now, what's not really considered in these kind of mathematical arguments of 3 million to 1 or billion to 1 is that many bacteria, many common bacteria, express identical A and B antigens. Now that they are found on the surfaces of human red blood cells, same exact molecules are found on many bacteria. For many of these objects, significant contamination has been described. I think it's something that you have to take into account when you're trying to make connections between these various objects. The types of studies that were done were serological tests using antibodies. Antibodies cannot distinguish between bacterial AB and human AB because, again, the molecules are exactly the same. There are ways you can distinguish this using DNA methods, but those have not been done. There are some other issues with AB typing that I'm really not going to get into apart from the ones that we just talked about. But it should be pointed out, even if the AB typing had no issues at all, shared AB is not scientifically sufficient to demonstrate the claims of a single origin among these various objects. To do that, you would have to use markers that are unique among individuals. So some examples of that, you could use nuclear DNA or mitochondrial DNA regions. This is typically done in standard forensic analysis. You could also use HLA transplantation antigens. What HLA refers to when a person goes in for a transplant and they get typed to see if they're a suitable donor or recipient, they're looking at their particular HLA profile. HLA has somewhat of an advantage over the other two in that it gives you options. You could test that at either the DNA level or the protein level. So if the DNA is not that great of a shape, you could look at it at the protein level. Proteins are a lot tougher. They tend to stick around a lot longer. So here's the references for these. The top one, this talks about the shroud, relics and Eucharistic miracles related to AB typing. The bottom one talks specifically about the shroud and AB typing. So to kind of sum this up, this is my own opinion, 2025. I would have to go, based on the kind of tests that we talked about, I would have to go not determined for all three of these. Now, I believe it's important to realize that most of these studies that were described, they were works in progress. They never really made it to that depth and completeness that you would need to publish in a peer-reviewed scientific journal, which none of those studies ever did. Going forward, my own opinion, I would not worry about validating that the blood is from a male. I would not worry about the AB. I think it's a scientific rabbit hole. The HLA is vastly superior for that kind of thing. I think that the one to particularly focus on characterization is human. It's a fundamental characteristic. Now, is there other evidence to indicate that the blood on the shroud is human? Of course. Are these type of scientific tests still important? Of course. Scientists are going to want it to be demonstrated as thoroughly as you can. There was nothing a matter with the approach that was used. It's just the tools that were available back then didn't really have that discrimination that they now have. So that's all I'll say about that. We're going to now go to part two. We're going to talk a little bit about blood transfer. So here we have the front side of the cloth, which is in contact with the body, and then the reverse side. Here's the blood stains on the front side on the left. So this is what we're kind of typically used to looking at. Here are the blood stains coming down the arm, and here's the side wound. Over here on the right, this is the reverse side, which you really don't see that often. But you can see here you have the blood stains coming down the arm, and here's the blood stains in the side wound. So many blood stains soak through to the reverse side. Well, how was the blood transferred? So there are several schools of thought on this. Well, one is that the blood was transferred normally, like blood typically is, when it's in a liquid or semi-liquid state. And I'll admit this is the one that I personally kind of lean toward. But others have pointed out, well, Christ's body was probably on the cross for a bit before they took it down, or there are other blood stains on there that would have dried. So is there any way that dry blood stains could have transferred to the cloth? Well, the scriptures tell us that Christ was buried in a cave tomb. We really don't know exactly which cave it was. A lot believe it was the Church of the Holy Sepulchre. That certainly in the Catholic tradition. But one thing that caves have in common worldwide is their low temperature and high humidity. And measurements have been made around the Church of the Holy Sepulchre in that cave area, which fall within these parameters. So I wanted to do some experiments to kind of look at this idea. So I took blood, I added it to skin. I use pig skin here because that's as close as you can get to human skin. It's what medical students use when they practice how to learn sutures. I put blood down on the skin and I let it dry really well. Just to verify it was dried, I put a piece of material over it to make sure nothing transferred. If that was okay, then I put a fresh piece of material on top of it. I put this inside of a humidity chamber. So on the left-hand side, you have low temperature. On the right's room temperature. I did that to kind of simulate moving toward the mouth or the opening of a cave. And I put this down, and then I took it off overnight, so after like 12, 14 hours later. So with the low humidity, you really don't see any imprinting. You only see it under the high humidity conditions. Here's one where you're looking at the contact side. This is all under high humidity. And here you have it on the reverse side, so you can see the blood soaks through to the other side, just like the shroud. Here's a cluster of blood stains. So here's the original that's on the skin that's dried. Here's a control transfer just to verify that it's dry. This is the imprint that you get the next day, so you can see it pretty faithfully reproduces the blood stain pattern. The edges are also relatively smooth. Here what I've done, I've kind of tried to mimic some of the more prominent, well-known blood stains on the shroud. So here's the epsilon blood stain. Here's the original on skin. Here's a control just to verify that it's dry. Here's the imprint the following day. I got some cracking on these. I think it's probably because when I pulled these off, I sort of folded it in half a bit. But you can see it pretty faithfully reproduces the original stain. Also, the edges are relatively smooth. Here we're looking at some wounds on the upper arm and the wrist. So the one on the upper arm, I picked this one because that's been suggested to have certainly been dried. So here's the resulting imprint that I got the following day. So it's pretty pristine looking. The edges are quite smooth. We go down to this wrist wound. This one's particularly interesting because Miller and Pellicore, members of the original STIRT team, described that when they looked at this under ultraviolet, they saw these fluorescent halos or edges, which was subsequently shown to be serum. So I'm going to kind of look at that. So here's my original on skin. You really can't see this that well under visible light, but here's the serum right here. If you look at the imprint the next day, this is under ultraviolet, you can see here's the blood stain, and here's the serum. Transfer is pretty neat right here. That is, everything kind of stayed in its own lane. So humidity imprinting demonstrates that dry blood stains are effectively remoistened and transferred to the cloth. Now, some have pointed out, well, there was probably a lot of blood on the body. So if this is really going on, you'd expect to see a lot more blood on the shroud than you do, which is certainly a very reasonable point. I'm just kind of throwing this out there as a possibility that some of the blood stains may have been transferred that way. It's just kind of food for thought. Was the body washed prior to placing it in the shroud? So I'm going to talk just a little bit about blood clotting, and then we'll come back to the question. In the late 1990s, Dr. Gil Lavoie, in his first book, he showed some classic blood transfer experiments where he put blood down and let it dry, and then after a certain period of time, put linen on top of it. So I wanted to extend these in two ways. Number one, I wanted to bring in the serum, the ultraviolet to check out the serum and to see what was going on with that. Number two, probably most importantly, I wanted to compare this to what it would look like if you added blood directly to the cloth. Let's say how a forger might have done it. So here you're looking at blood imprints. This is all under ultraviolet light. So when clotted blood is transferred, you get this fluorescent serum halo or edge. So what happens when blood clots, the clot retracts, kind of pushes the serum to the outside. You can capture that in an imprint. If you put blood directly on the cloth, no halo or edge. Now what I've done here, I've tried to add blood in a variety of ways, just directly to the cloth. I dropped it on, I used a piece of wood, I used metal. Here I use a cloth applicator, no halo edge. Only time you see it is when clotted blood is used. This top guy right here, what I did, I let the blood clot. Then I stirred it really, really fast and really, really hard to break the clot up. I added that to this is filter paper, added that, no halo. This one down here, I let the blood dry for several days. I ground it up in a mortar and pestle. I then added water to that, transferred it directly, no halo. Now, I did go on to show that there are ways you can get halos without blood clotting. So one way you can do this, you can use various natural additives. They are naturally fluorescent, so you can see it that way. Another way, you can add blood directly to the cloth, so no additives, but you can add it directly. And then just through a pretty mild heating for a short period of time, you can see the halos that way. Now, my own opinion, I don't believe any of these was involved with the shroud, because I think if it had been done this way, you would pretty much see it on every single blood stain that's there, which you don't. Now, Alan Adler did make the statement one time, well, every single wound shows a serum halo around it. He said that after looking at photographs. It's clearly an exaggeration. It's not what Miller and Pellicore in their firsthand observation of the shroud described. It's not what they wrote about in their paper. They said many show this, but certainly not all. You can also look at the photos on shroudphotos.com, and you can check it out. So, my own opinion, I think the most scientifically logical conclusion. Serum halo edges indicate that clotted blood was transferred to the cloth, which is a bit of a game changer because it indicates that a body was involved. Was the body washed prior to placing it in the shroud? Serum has an extremely high water content. It's about 92% water. So I would believe that if the body had been washed, these serum proteins would have dissolved in the water very, very well and would have been washed away. They wouldn't have been available for this imprinting that we see. So to look at this experimentally, what I did here, I put blood down on glass, and under the UV, you can see the halo. And then I did a rainwater rinse. So I used rainwater because I thought it would be more similar to what they might have used back then as opposed to, say, tap water, distilled water, something like that. You can see after the rinse, the way I did this, I didn't touch the blood stain at all. I was like 6 to 8 inches away and did a very small spritz, very small volume of water. You can see even with that, the halo is completely wiped out. This is on skin, so here's the halo on skin. Before the rinse, after the rinse, it's gone. This is isolated serum, nothing but serum, pure serum. Before the rinse, after the rinse. This is on skin. Here's the serum, before, after. Now, some have suggested, well, maybe it was kind of a speed wash thing, that, maybe they kind of wash real quickly, something like that. So I did the same kind of spritzing, and then I did an overlay to make an imprint. You can see it's pretty smeared, pretty smudged, not like what you observe on the shroud. Certainly not what you see with something like the humidity imprinting where the edges are relatively smooth. So I believe these results are consistent with the idea that the body was not washed. Now, probably the greatest proponent of body washing was Dr. Fred Zugaby. So he believed that the body was thoroughly washed, and the blood you're looking at really is blood that is oozed out from those post-mortem wounds. It was noted as far back in the 1800s that normally blood is spontaneously coagulable, that is, it clots. However, capillary blood of a cadaver is always fluid and incoagulable. The blood in the veins of the limbs is more often fluid than not, is only exceptionally coagulable. In the 1960s, it's well known that the blood in sudden death, asphyxia, or shock shows fluidity but no coagulability. In Zugaby's own firsthand observations of the cadavers that he washed and showed that blood, in fact, can ooze from those wounds, but in his own firsthand observations, he said after post-mortem blood flows from a wound, it rarely coagulates, but it may dry. It dries through simple evaporation. This seems at odds as to what you observe on the shroud, where you have these fluorescent halos that are indicative of clotted blood that's transferred. So I wanted to look at that experimentally. Here's control blood. This is under UV, so there's the halo. This is blood that's been treated with anticoagulant. No halo is present. Here's the same experiment on skin. So here's the halo that you see in the control. If you impair coagulation, there's no halo that forms. There have been studies that have described how the pH of blood drops hours after death. It starts to show a little bit of decrease in pH. So I adjusted the pH of the blood to match those conditions. Here's the control with the low pH. Here's the halo that forms. This is low pH, and when coagulation is impaired, there's no halo that's present. Now also I want to make some imprints of the blood. So I put the blood down and then put a piece of material on top of that and made an imprint. So here's the control. The blood's clotting. Here's the serum halo or edge fluorescent that you see. When you impair coagulation, it's not present. So these results show that ineffective coagulation, it's associated with the lack of formation of serum halo rings or edges. I believe that these data don't really support Zuckerby's idea that body washing with successive oozing from wounds was a major transfer mechanism for the Shroud bloodstains. Now, I want to finish up with this. Quite a few places on the Shroud, you'll see a blood stain and then a serum halo edge, and then you'll see a water stain right on top of it. We've talked about how serum has extremely high water content, about 92%. So it seems kind of at odds how you could have a serum halo edge and then water right next to it. You'd think it might have wiped it out. So to look at this, I took some serum, put it on a piece of filter paper, and then I divided it in half. The control half I didn't do anything with. The other, I exposed to various solvents. Water's the most important, so that's what I'm going to show. So if you look at the top, this is under UV. This is the control half, and this is the half. This is fresh serum that's been dried, and when you expose it to water, it's wiped out. Pretty much what you would expect. If you look at aged serum, you'll notice two differences. Number one, there's a turbo boost in the fluorescence. Number two, it's water insoluble. It's not dissolved. Now, the aged blood used in these studies are aged serum, aged blood both, about 2 years old. You really don't have to wait 2 years to see this. You can see this at about 6 months. The serum undergoes a structural change where it becomes more fluorescent and also water insoluble. I'm currently looking at the molecular basis behind the change. So these results demonstrate that aged serum stains show increased fluorescence and water insolubility. And the second part, in particular, the water insolubility observation might help to explain how serum edges persist on the Shroud in regions that were affected by subsequent water stain. Through somebody tossing water on the Shroud, say to put out a fire, or maybe the Shroud was stored in a clay jug during its history and the water got on there that way. Thank you. Yes.
If you're familiar with the Sudarium,
yes.
has that been tested in more scientifically rigorous?
No, sir. So pretty much the same. Actually, I don't want to say less rigorous, but maybe not quite as much as the Shroud, but the same kind of test, same kind of thing. Okay. Yeah. Yes, sir.
Thank you for your lecture. I was wondering over here on the left side. Oh, were you still talking? I couldn't hear it over here. Joe handed me the mic. Go right ahead. Sorry.
Thank you. The second question is, you said the pH level changes.
Yes.
Are there other indicators if something is postmortem blood?
So with fresh blood, you can do that. With blood from stains, it's difficult. It's difficult to do it, and it's a very good question. My own opinion about this, I think when you look at the blood stains as a scientist, you have to be willing to separate them out from the image and just look at the blood stains themselves. And then I think you really, even as a scientist, you have to take that and separate it out from the gospel narrative. But we know this is post-mortem because, after that kind of thing, just look at the blood stains themselves. I think it gets difficult. So you could maybe try to do that colorimetrically, but even working with pre-mortem blood, depending on how it dries, the density, you'll get variation. And you would have to make that convincing, I think someone would have to use blind samples of pre-mortem and post-mortem blood, and well, how does this sort? And then they show you the results and you match it up. So it's not an easy thing to do in my own opinion. And even with the halo rings, so when you see them, you know that you have clotting, that the clotting's happened, but you can have clotting and not have a ring. Something that's often not appreciated, especially on a body where it might be moving or going around a contoured area or something like that. If you see the edge, the fluorescent halo or edge, yeah, you know that the blood's clotted.
Last question.
Yes, sir.
There's been some claims because when you match up the stains with the image, that a trained eye can see if it's arterial or if it's venous. Do you agree?
So I'm not really a pathologist or a physician, but I'm personally not really convinced. But that's just my own opinion, and I'm not trained in that particular area. Yeah. Thank you. Okay.
Thank you for your lecture. Do you see presence of pulmonary edema, especially in the side wound?
So a lot of times when people look at, so pulmonary edema is basically just going to be serum that's essentially diluted. It's where all the fluid comes from. A lot of people will look at the side wound and see those spaces in there and say, well, it's blood and water because blood and water came out. Blood and water, blood and pulmonary fluid, blood and serum, they don't separate like this on this. It will mix together. So that's likely just a gap, maybe where it didn't transfer or something like that. Yes. And it's not just, I've asked 15 plus people that are pulmonary experts, and that's what they say. And I can tell you, I've tried to mimic pulmonary edema by diluting out serum and mixing and stuff. You don't see that kind of separation because it's all blood, serum, all very, very water heavy, very, very water based. Yes. Yes. Yes.
Interesting presentation.
Thank you.
We see the chest wound, and thank you. In correspondence of the chest wound, we see the serum edge, but only in one part. If we look, for example, to the bloodstains more lower, there we see no serum wound. Is it possible to think that some bloodstain were formed on the body of Jesus before his death, like that of serum, but other bloodstains, like those on the bottom, were formed during the wrapping in the sepulchre?
Yes, absolutely. It's my own opinion, but absolutely. I don't think it's kind of all or none. I think it's probably my own opinion. I think it's a little bit, yeah, of each. And I think you would certainly have, pre-mortem and post-mortem on there. It's just trying to sort out objectively. Yes, absolutely, yeah.
Thank you.
Yeah, yes, sir. Thank you. Yes, sir.
Yes.
Can you clarify forensics? They say that to test blood, a blood type, as blood type ages, as the blood ages, it shows AB that you cannot actually determine the type because of the age. Over.
Okay. So there's two ways you can type the blood. So you can type what's on the red blood cell. That's called forward typing, using antibodies. That's what I showed here, talked about here, okay? And that's what, if you go to get a blood test, you're gonna donate blood, you're gonna put it, that's what they do there. But before they're ever gonna put blood in someone, they're gonna use another type of test because they cross-check each other. So the second type of test is called reverse typing. So they don't look at what's on the surface of the red blood cell, but they look at what is in the serum. So if a person is type A, they will have antibodies in their serum against type B because they've never seen it. If a person is type B, they will have antibodies in their serum against type A. If a person is AB, they will have no antibodies against those in their serum because they've seen both of them. If a person is type O, they will have antibodies to both A and B in their serum. So those types of tests are functional. The antibody has a strict three-dimensional conformation. It has to have that in order to recognize the antigen, okay? Over time, with age, that falls off. That functionality decreases. So when you go to test this, if you test an age sample, we don't get a result. Well, was it originally AB? Or was it just A that with time, the functionality fell off? B, with time, the functionality fell off. O, with time, fell off. They all meet in the same place, which is why reverse typing is typically never used for age samples. BIMO will only put it in there, but when it's AB, it's a catch-22. It's just a circular argument. You don't know if, well, it truly was AB or it was one of the others that with time, because that's a functional test, it fell off. With fresh blood, it's no problem. And they'll agree. You can tell both tests are going to match up. Yeah, it's a very good question. Yes. Yes, sir.
I'm missed the front part, I'm sorry. Did you talk about some Shroud, some of the Shroud research has shown that the type was AB negative?
So the negative, what the negative part refers to is called Rh factor, Rh from a rhesus monkey. That has never been tested on the Shroud. So I've talked to Byman Baloney and asked him about it, and others. That has never been tested. So you'll sometimes see that, well, the blood is AB positive. So to immunologists, that means type AB, Rh positive. But someone else, well, that just means it's AB, because it's AB positive. But the Rh factor has never been looked at in the Shroud samples, but that's, will be out there.
Some researchers had called out?
Yes, yes, I've seen it on both sides, all across the board. You bet. Yes, ma'am?
Can you address, I think Dr. Fanti addressed it, that the blood, they've determined that, the blood that they determined that in fact came from a man that was tortured? Have you done studies on that
I have
also?
not done those particular studies. I am, in my own opinion, that would be still up for interpretation. And that's, I never look at this stuff and say, well, okay, I mean, I do look at it and say, what do I think? But I will always contact as many people as I can in specialties and show them it. And a lot of times I don't even say it's from the Shroud and say, tell me what do you think about this and that? So for me, I can't really make that claim. I can speak to the bilirubin part, which I did look at. So one hypothesis, this is Alan Adler's, that the blood stays red because of high bilirubin. So I intentionally loaded up blood with extremely high levels of bilirubin. In my hands, it did not cause it to stay red. I've looked at rats that genetically have high levels of bilirubin because of mutation. That blood did not stay red. Bilirubin shows a unique UV signature. It's a very unique fluorescent signature. That type of fluorescence is not observed on the Shroud. It's also extremely light sensitive, so it's kind of hard to understand how that may be maintained over time. But as far as torture, that's kind of the only thing I personally experimentally have done. Yes, ma'am. Yes, sir. Tristan, how are you?
Tristan.
Thank you.
Regarding this question about bilirubin
Religious question?
yeah, are you aware of the study done by Paolo Di Lazzaro about bilirubin with UV light?
Yes.
And what do you think of it?
So I love Paulo to death. I have tremendous respect for all that he's done. But my thing about that, when he shows that the reddish is maintained, it's through a RGB value, a computer value that's plotted on a graph. When the reddish color has been described for centuries, it's visually by eye. And I understand that you can move it from one place to the other and you get a different color interpretation. Take the control along with it, it's going to be lit up under the same exact pathway. So I'm not really convinced with it. If it were more visual and stronger, yes, but I don't think the differences on that are that huge. So that's my deal with that. But loads of respect for Paulo, big time. Yes,
We have
ma'am.
about 5 minutes left, so
Okay.
maybe one or two more quick
Yes, ma'am.
questions.
With regard to, you were talking about how blood, or how the serum does not become insoluble
Right.
until about perhaps 6 months.
It's around
or
6
so.
months, yes.
So as soon as you said that, it caused me to think in terms of the image-making process, which I've studied the yellowing of cellulose, and I mean, I think that some sort of energy, which I think the energy of the resurrection created the dehydration and the oxidation of the cellulose that gives us the straw yellow color for the body image. And so I was thinking in terms of that energy, couldn't that work in the same way that people do the artificial aging of linen to create the age and the brown and the coloration? And could not that artificial aging from perhaps some sort of heat or energy from the resurrection or whatever, could that then create the insolubility in terms of speeding the aging of the serum, do you think?
So these samples I showed, they're not from Jesus. It's just ordinary serum. So it shows that property of increased insolubility and increased fluorescence. You can speed it up by heating and artificially aging it. So I've done some of that with Sam Pellicore, original STERP team guy. You can do that. I don't think it's really related to the image process per se. I mean, I think it's a separate thing, but just normal serum shows that. It's a similar kind of thing when you're tested for diabetes. What happens is hemoglobin gets modified with carbohydrates, and there's an antibody that recognizes that. The carbohydrates are present in the serum. So I'm working on this now. It's not come out, it's not finalized, but it's probably a similar reaction. It's Maillard type reaction is what causes it, but even just normal serum will do that. But I do think, related to the shroud, I do think the reason that fluorescence kind of pops out is because it's aged. When it's aged, you're going to be able to see it a lot better, but that also depends on what the background is. But so even normal serum will do it.
All right, just one or two more quick questions. This gentleman here, and then I think you had your hand up, will be the last two.
Thank you so much for your work. I really
Thank you.
appreciate what
Thank you.
you've shown us today. And so you've specifically talked about the fact that, well, we can't determine that it's human blood or it's male or that it's AB. Can you tell us?
The tools are there to do it now.
Right,
Yes, sir.
right.
Yeah.
But can you then confirm for us that it is blood and that what is the confidence level on that?
So you can, I think you look at the test, you can look for paint pigments, et cetera, you don't see it. You want a straight shot to show it's blood, you go for hemoglobin, signature molecule. Several different ways. Spectroscopy, chemical tests have been done. Okay, so is it blood? I would say you bet. You know? So then, what type of blood? There you're gonna go immunological test. So these tests never nailed down the species, but they did show, well, you've got immunoglobulin and albumin, two major blood proteins. Boom. How do you immunologically get a positive result without it being blood there? I would say you don't. So I would say you bet. Yes, sir. Yes, sir.
A bit of a follow-up on Professor Fonte's question. So the blood that's outside the area of the image,
Okay.
I'm thinking particularly where the forearms are,
Yes, sir.
toward the edge.
Yeah.
Can we say anything definite about its characteristics versus those that are within the area of the image? And then also, what's our best guess at why the blood has remained red?
So I would say that depends on who you ask about that one. And I think, so my own personal experience, Rogers proposed it with saponin. I've tried it. In my hands, it didn't do it. Adler, bilirubin, I've tried it. In my hands, it didn't do it. Apollo's shown some stuff with the photons and then the high bilirubin. But again, I would like to see it visually do the change. And I think there could be other ways. I'm working on it right now. I don't want to say too much, but I think there are other ways that could get you there. Yeah, and it could be some combination of things, but yes, sir. Good question.
Okay, that'll be it for now. We have mass from 12 to 12:30 and lunch from 12 to 1, and we'll meet back here at 1 o'clock.